We're about to get started with the next session. Can I just, we're about to get started with the next session. Can I just remind people who want to attend the keynote that everything which is being broadcast in here is also being simulcast out to the East School. So if you're unable to find a chair in this room, there'll be lots of overflow seating. There will definitely be a chair available for you in that room, and you'll be able to watch the keynote there. Before Helen Barron's keynote, we're going to have a brief presentation from the BBSRC. So we've been very grateful this year to receive some funding from the BBSRC, which are one of the funding agencies in the UK. They form part of UKRI. And we've been particularly grateful as a CCN community here in the UK over the past year, because they've not only funded this conference, they've also had a much broader £2 million funding call that was specifically targeted towards seed funding in cognitive computational neuroscience. And the first 10 projects that were funded by that had a little satellite meeting over at Wadham College yesterday, and the kind of architect behind that at BBSRC has been Kerry Wynne Thomas, and she's going to tell us a little bit about the funding opportunities that are available within UKRI at the moment. So let's welcome Kerry Wynne.
Lawrence, thank you very much. I'm being credited with being an architect behind something like that is very generous of you. But yes, hello everyone. Thank you very much for having me here. I won't keep you long before the keynote. As Lawrence said, my name is Kerry Wynne Thomas, and I'm here representing the Biotechnology and Biosciences Research Council. And as you can see from this really quite persistent graphic, this forms one of 9 bodies or research councils that are operating under the wider umbrella of UK Research and Innovation, UKRI. And UKRI technically is known as a non-departmental government body, and allegedly we do the behest of government. We do their bidding in terms of funding research and innovation in the UK, and we allegedly do this sensibly and efficiently. And we have a budget of about 8 billion that goes to fund research. And as you can see from the Research Council's names that keep cropping up, we have a very broad research remit, so we effectively take care of all research and innovation that goes on, or most research and innovation that goes on in the higher education sector within the UK and across strategically funded institutes. So that's who we are. And I thought that I would share with you today some strategic documents in the hope that they will be useful and relevant to those of you in the audience, but also because we spend quite a lot of time navel-gazing over these documents internally, and then we always end up being quite disheartened when we find that no one in the community has had time to read them. So I thought that I would summarise some of them for you now. So UKRI published its current strategy, which extends to 2027 last year, and this really sets out our very high-level priorities for delivering the UK's vision and research agenda. And in it, we delineate a number of strategic objectives through lots of lurid vector graphics and stock photos, but also within this document, we outline a number of our strategic themes. And these are very high-level strategic themes that span the entirety of UKRI's remit. And probably at first glance, it looks as though there's very limited alignment to what's covered in the research that's being presented at this conference. But actually, I would make the argument that probably a lot of the work that's being presented here does fall within alignment with some of these strategic themes, particularly securing better health, ageing and well-being, but also building a secure and resilient world. So this is a lot of the high-level language that we use that really encapsulates the research and innovation that we aim to fund and support. And the reason that I thought it would be pertinent to share is that what we often recommend to people who are applying to UKRI, whether that's for grant funding or a fellowship, any kind of funding scheme, we like to see alignment with these overall strategic themes. And so if there's any way that you can crowbar your research into one of these topics, or rather to demonstrate alignment with them, then we always recommend doing that. This is very easily findable online, and as I say, it's always worth having a read through. A few years ago, UKRI also published its own review into AI and the role that it will play in transforming the research landscape. And again, this is another very useful and very easily Googleable document to read if you are working in this particular space and you're interested to know how UK government and UK research and innovation are going to be supporting both discovery-led research but also application-driven research and what the key priorities are in this area for us and where we want to see alignment in different science areas, including those that I've listed here. So again, another very useful document to read if you are applying for a research grant with any of the research councils and the work happens to intersect with AI, and this goes for BBSRC as well. What I'm going to do next is just summarise some upcoming calls. Again, hopefully some of these will be pertinent to those of you sitting in the audience, and so if you haven't yet had a chance to peruse the UKRI funding finder recently, then hopefully this will be useful for you. So we very recently closed, actually rather annoyingly, but a partner call with international researchers working in AI for bioscience, and so that gives an example of some of the things that BBSRC are currently funding in the international landscape. We also have responsive mode funding, which is the primary mechanism that we use to fund neuroscience research through BBSRC. Next closing date for that is the 27th of September, and I'm sure many of you in this audience will be intimately familiar with that research mechanism. We also have a number of spotlight areas, although nothing on neuroscience yet, although that may change in future, so please do keep an eye on that horizon. We've also got our Transformative Research Technologies call, which is, it provides funding for the development of research technologies with transformative potential in the biosciences and can be used for the purchase of various forms of equipment. So that might be useful if you're working in a lab and are interested in buying something new. UKRI also have a number of AI-related calls at the moment, so we've just launched our Impact Accelerator Round 1. The closing date is on the 7th of September. And again, this can be used to support sort of interdisciplinary and translational research and knowledge exchange, looking at the responsible and trustworthy use of AI, if that's relevant. We also have an international partnerships call coming up in this space where you can have funding to develop partnerships with organisations around the world, again looking at the use of and the responsible use of artificial intelligence. Closing date for that is on the 14th of September. All of the research councils, I'm aware that this is obviously an international conference, routinely put out calls for international partnering awards, and BBSRC also does that. And these awards can be very useful if you have collaborators either based in the UK or you're in the UK and you have collaborators internationally. They can be used to secure travel and funding for other events like workshops, and so they can be very useful pots of money to apply for. We don't have anything forthcoming at the moment, but I know that this is on the horizon. And so please do keep your eye out. And of course, it would be really difficult to summarise all of the myriad calls that UKRI and BBSRC put out every year. But the best way that you can keep up to date with those, if you haven't yet seen, is UKRI's funding finder, and I'm pleased to say that it's just as brightly coloured as all of our other web presence. But this is a really, really useful website that you can access through the UKRI main site. And it can be filtered by research council subject area, but also if you happen to be based internationally and you're interested in a number of the sort of lead agency agreements or bilateral funding agreements that we may have with other funding bodies around the world. So this is your one-stop shop to keep a weather eye on the funding horizon as far as UKRI goes. And then very quickly, just finally at the end, apologies, this is a bit of a noddy slide, but we've just recently launched a BBSRC-funded network known as the AI Bio UK network. They haven't actually launched yet, so I've not been able to pilfer a nice logo or a website link. But this network was really created with a view to build a much stronger community profile for AI within the biosciences in UK, so a little bit broader than the remit of this conference, but it's also to facilitate networking and knowledge exchange and to support greater sort of AI awareness and education, but also training across the research communities that are involved in this kind of research space. Very useful for early career researchers, and they will have some money to fund small-scale pilot projects as well. So once they come online, please do look at the funding finder and follow along and become a member if it's relevant to you and your work. So that's all there is from me. So I will hand over now to make sure that we can start the keynote. And thank you very much for your time, and thank you to Chris and Lawrence for having me speak.
Thank you very much for that. I have the honor of introducing our next keynote speaker, Dr. Helen Barron. She did her undergraduate at the University of Cambridge and her PhD at University College London. In 2015, she was awarded a Junior Research Fellow at Oxford here, and recently, in 2022, was awarded a UKRI Future Leaders Fellowship, which has allowed her to start her own team running at the MRC Brain Network Dynamics Unit and the Wellcome Center for Integrative Neuroimaging here at Oxford. Her research focuses on memory, representations with a really impressive multi-level toolkit and a rich interdisciplinary perspective and approach that not many single labs offer in one go. So rather than me telling you more about it, I'll let her tell you about it. Please join me in welcoming Dr. Helen Barron.
Thank you very much for the kind introduction and the opportunity to talk here today. It's really nice to be here. So I'm going to be talking about how we might build cognitive maps during periods of rest and sleep. Okay, so you might imagine that there are 3 different stimuli. You can think of them as visual cues or auditory cues, X, Y, and Z, and each of these are represented by different ensembles or assemblies of neurons in the brain. And when we form associations between, say, X and Y, we learn to pair those 2 cues together. Through Hebbian plasticity, we can get strengthening in the synaptic connections between those 2 ensembles of neurons that represent those cues. And same goes if you learn to pair QY with QZ, you can imagine a situation where you then strengthen the synaptic connections between Y and Z. But what I'm interested in, and my group are interested in understanding, is what this might mean for unobserved links between different memories. So in this example that I've given you, the person has explicitly paired X with Y and Y with Z, but under certain circumstances, you can get a situation where you've got Hebbian plasticity also between X and Z. And that might occur due to the fact that you learn X, Y, and Y, Z in short succession, or potentially during periods of rest, if you reactivate X and Y and Z in short succession, you can end up with a situation where X and Z are firing in short succession from each other, and you get wiring essentially between those 2 ensembles of neurons. So this kind of process is really interesting to me because it effectively allows the brain to go one step beyond what it's learned, beyond reality, if you like, and allows for coactivation between different memories that haven't actually been experienced together. And so from a behavioral point of view, we can think of this process as potentially underlying the formation of, or the inference of a shortcut while we're navigating. So this example kind of builds on ideas from people like Tolman in the 40s, who showed that when rats are navigating a maze, if they learn this blue route here, they can subsequently infer that there's a shortcut in this maze that allows them to get out of the maze faster than the route that they've actually learned. So forming these kind of shortcuts may allow us to sort of facilitate highly flexible behaviors. And so what we can think of these shortcuts as being as sort of shortcuts in our kind of mental or internal map, or what people refer to as a cognitive map, our representation of the world that is stored in our brain. But of course, there are situations where it may be suboptimal, actually, to form shortcuts between different memories or links between memories that haven't been experienced together, because by definition, this linking of different memories is taking us beyond what we've actually experienced. So by forming these shortcuts, we might actually distort our internal map of the world relative to what's out there. Okay, so today I'm going to talk about 3 studies, the first of which is published already, and this one's really asking how we represent inference in the brain, and I'm going to show you that inferred relationships can be represented by spiking activity in the hippocampus during periods of rest and sleep, and this study was actually performed in humans and mice, but the key finding I'm going to show you is from mice. And then I'm going to show you 2 unpublished studies in which we try and manipulate reactivation or replay in the brain, and then ask, if we enhance replay, does that then facilitate our ability to make inferences and form shortcuts in our kind of cognitive map? So I'll go through these step by step. So the first study is about when and how inferred relationships are actually represented in the brain. So this was a study that we conducted in both humans and mice, and we tried to take advantage of the different tools available in those different species in order to try and tackle this question. So first of all, we developed an inference task in both humans and mice that allowed us to investigate inference. We then looked at where in the brain inference is or what's happening in which areas of the brain are actually necessary for inference, and I'm not going to actually show you that data today. I'm just going to tell you that if we use causal manipulations in mice, if we impair the hippocampus, we see a deficit in terms of their ability to infer. And instead, what I'm going to focus on is the computations that are occurring in the hippocampus during inferential choice, and also talk about what's going on offline during periods of rest and sleep. So to start with the task, the sort of structure of this task is relevant throughout the talk, so I'm going to just go through it carefully with you. So first of all, in the first phase of the task, our subjects learn to pair what we call stimulus X, which is an auditory cue, with stimulus Y, which is a visual cue. And then in the second phase of the task, they learn that the visual cues are paired with either a reward, which is if it's humans, we use a monetary reward. If it's mice, it's a sucrose reward, and the mice are food deprived. And then in the second set of cues in the green set down here, this reward is neutral. So this is our kind of control condition here. And for humans, that's a wood chip of no value. And for mice, that's a water drop. And the mice have free access to water. So that water drop is effectively of no value for them. And then in the final phase of the task, we have an inference test where we play the sounds, the auditory cues X, in isolation, and then we assess the subject's ability to infer the appropriate reward in response to that auditory cue. And the environments look like this. So we used a box here for the mice that they're running around in, the visual cues appearing on the walls, and then auditory cue played in, and then the drops of sucrose or water are delivered to a dispenser here. And then we try to match this environment using virtual reality in humans. So we, where possible, kept things as similar as possible, but we had the rewards, the monetary coin and the neutral wood chip appear in this wooden box in the corner. Okay, so then we have the following kind of structure. In both species, we have the observational learning, the auditory to visual cue pairing, and then the conditioning period across multiple days. And I'll just go through this bit on the bottom because it's relevant later. So in terms of how we measure inference, in mice what we do is we play the sound into the environment, and then we're going to look at whether they seem to show any reward-seeking behavior in response to that sound. So to do that, we're going to measure how long they spend in the area around the dispenser after the sound has played. In humans, what we do, we have them in the scanner at the time when they're performing the inference test. We play the sound while they watch a video of the VR environment, and then we ask them whether they'd like to look in the box where they previously have found the coin. And so we use those two measures as a measure of reward-seeking behavior. And so we have two phases of the task where we're sort of getting a readout of behavior. The first is the conditioning phase. And in both humans and mice, we see we've trained them to show this strong conditional response to the, what I'm going to call the orange visual cue compared to the green. So here you can see their reward-seeking behavior on the y-axis, and you can see they're more reward-seeking for the orange visual cue set 1 compared to the green visual cue set 2. And in gray, I'm showing here bootstrap distribution of the difference between set 1 and set 2, which you can see is highly significant. And same goes for mice. So in general, the mice behavior is more noisy. They haven't been explicitly instructed on what to do in the same way that humans have, but nevertheless, they're showing a highly significant conditioned response here. And then the more interesting bit happens during the inference test, where we're playing the auditory cues in isolation, and then we're going to, again, assess how reward-seeking our subjects are. So Humans are very good at this, despite the fact that we actually made the task a bit more complicated for them by including additional auditory cues. They show this highly significant inference response, showing more reward-seeking behavior for orange auditory cues compared to green auditory cues, as if they're expecting the reward here, but not here. And for mice, again, the behavior is more noisy, but you can see that they are showing more reward-seeking behavior for the orange auditory cues compared to the green. Okay, so as I said, I'm going to miss out the sort of, just in the interest of time, I'm going to miss out the section on how we try and work out which brain areas are involved in supporting inference, and I'm just going to tell you that the hippocampus is necessary for inference, and we determine that using some optogenetic manipulations in mice. And instead, I'm going to jump to the section on the computation that seems to be happening during inferential choice. So I'm now going to look at what's going on in the hippocampus in both humans and mice. And so we wanted to find a method that would allow us to look at data in different modalities. So in humans, we have fMRI data, and in our mice, we have electrophysiology data, recordings from single units. And so we decided to work with representational similarity analysis developed by Nico Kriegerskotter and really nicely demonstrated across humans and macaque in this paper and subsequently in computational models as well. So regardless of the type of data that's being that the method is applied to, you can extract out what the underlying representations might be. So how does this work? So the idea is that you look at the activity pattern, whether that's across voxels with fMRI or across neurons with electrophysiology, in response to stimulus 1, and you compare that to stimulus 2 by correlating those two matrices with one another, and then you can put the correlation coefficient into your representational similarity matrix here to indicate how similar those two representations are to one another. And then you do that for all your different stimuli in your task, and you build up a representational similarity matrix which indicates to you what the sort of structural content of the information is being represented by that particular brain area. Okay, so we're going to apply this method to our fMRI data acquired in humans at 70, and then our electrophysiology data, which was single unit recordings in mouse dorsal CA1 region of the hippocampus. And so we're taking the activity pattern across voxels for the different cues in humans and across neurons in mice for the different cues. And then we look at the correlation between those, and we build up our representational similarity matrix here. And what we were interested in is what's really being represented at the time of inferential choice. So at the time of inferential choice, we're playing the auditory cue to the subject in isolation, and then we're wanting to know what is that auditory cue eliciting? Is it eliciting a representation of the visual cue, or of the outcome, or both? And so if it's eliciting a representation of the visual cue, what we would expect to see is a high correlation between the orange auditory cue and the corresponding orange visual cue. So that we get, across all pairs of cues, we get this on-diagonal with a strong correlation coefficient or a positive correlation coefficient relative to the off-diagonal. And for correctly inferred trials, that's essentially what we see. We see a stronger on-diagonal compared to the off-diagonal, and that we don't see that for the incorrectly inferred trials. And that's the same for mice, where we have less cues, just two pairs of cues, or two triplets of cues, I should say. We see the strong positive on-diagonal for correctly inferred, but not incorrectly inferred. And so during inferential choice, what I'm showing here is that presentation of the auditory cue X elicits a representation of the expected Y, so effectively the learned association is being represented. And here this just quantifies this effect more thoroughly. So I'm now plotting here the difference between the on-diagonal versus off-diagonal, and comparing for all our recording days, and then comparing that with the null distribution where we shuffle the labels of the auditory cues. And you can see that for correctly inferred trials, this is significant in both humans and mice in the hippocampus, but not for incorrectly inferred trials. So what this is showing is that the hippocampus represents learned associations at the time of inferential choice. And then, based on this, we ask, is the learned temporal structure also preserved? So they've always experienced auditory cue X followed by visual cue Y in temporal succession. So is that temporal order also preserved, which you might expect if this is really a sort of prospective code that allows the animal to predict what's coming next. And so to test this, we looked at our single unit recordings in mice, and we first identified cells that seem to be preferentially representing the X cues, the auditory cues, and then cells preferentially representing the visual cues Y. And so to do that, we use a GLM to work out which cues the cells preferentially fire in response to. And then we identify our neurons, which I'm now going to call X and Y neurons, so auditory cue and visual cue neurons. And then we look at the firing relationship between them. And so on the bottom here, this is just a couple of example pairs of cells. So this one in particular is very nice. So this is a cell that preferentially fires for X2, the green auditory cue. And then on the y-axis, I'm plotting the probability of the Y2 cell firing. So this is the green visual cue. And you can see that at the time when the X2 cell fires, you see a big increase in the probability of the Y2 cell firing. And this is just the same data, but just now normalized to the baseline period from -50 to 0. So for that particular pair of cells, you can see that there seems to be clear temporal order preserved in the firing of those neurons. And if you look across all pairs of X and Y cells in the mouse dorsal CO1 region of the hippocampus, you can see that when the X cells fire here at 0, you can see that there's this significant positive increase in the probability of the Y cells firing. And again, this is just a bootstrap distribution of that effect. And if you compare that to cross-set neurons, so this is X1 to Y2 or X2 to Y1, you no longer see that effect. So it seems like this suggests that the learned information is really being represented according to the learned temporal statistics of the task. And so we refer to this as a sort of prospective code, which I think links in with a lot of the spatial navigation literature showing evidence for the hippocampus kind of prospectively predicting what's upcoming as mice or rats navigate around particular mazes. Okay, but I've shown you that when our subjects make inferential choice, they're representing the learned information. So in a way, that sounds a bit boring. And so where actually is the inferred outcome being represented? And we were actually quite surprised that we thought that the hippocampus would represent it, given that the hippocampus is necessary for inferential choice. If you opportunistically silence the hippocampus, you impair inference, and there's many studies by Howard Eichenbaum showing that if you lesion the hippocampus, you also impair inference. So where is the inferred outcome being represented? Well, to test this, we use our data from humans where we actually have a many-to-one mapping between our auditory cues and our visual cues, and our visual cues to our outcomes, and that allowed us to sort of disentangle this question. So in humans, we again use representational similarity analysis to ask, in response to our auditory cues, does the hippocampus represent the inferred outcome? And does it do something like this, which is independent of the intermediary visual cue, or does it represent the inferred outcome like this in a way that is dependent upon the intermediary visual cue? And we found no evidence for either of those two models. Both of them were not significantly different from null distributions, suggesting that the hippocampus doesn't actually represent the inferred outcome at the time of inferential choice. And this is despite the fact that we know from our data in mice that the hippocampus does represent these outcomes. It's not that it can't represent the outcomes, but at the time of inferential choice, it doesn't seem to represent the inferred outcome. So where else in the brain might represent the inferred outcome? Well, we used a searchlight approach to try and work out where or which other brain areas might be involved. And we found that there are two brain areas that significantly represent this particular model. So they represent the inferred outcome dependent upon the intermediary visual cue. And that's the medial prefrontal cortex and the midbrain, which we might think of as putative dopaminergic area. And this isn't just a value signal because we still get these two areas if we just constrain this to the neutral cues down here. And so I think then this raises interesting questions about how these representations of the inferred outcome actually get there, given that the subjects never have any direct reinforcement in response to those cues. So that made us think about what might be going on during periods of rest and sleep, or offline processing, in other words. Is it the case that during offline processing, the hippocampus is nevertheless able to represent the inferred relationship to effectively, potentially provide a training signal to other brain areas? And so just to introduce this idea, we know from a large body of work in rodents that the hippocampus represents particular locations in space, and that we refer to the cells that represent these particular locations as place cells. And so when an animal runs along a linear track, we expect there to be cells representing these different locations along the track. And so we can plot the sort of the firing response of this experience as follows. So we might have cell A firing, followed by B, C, D, E. And then what's really intriguing is that during periods of rest and sleep, we can see evidence for reactivation or replay of this previous experience. And this is occurring during these characteristic sharp wave ripple responses in the local field potential in the hippocampus, so the high-frequency oscillations that dominate the local field potential, particularly during non-REM sleep in the hippocampus, but these also occur during periods of rest, awake rest, while the animal is resting during the task. And so what we're observing here is a sort of temporal compression of this previous firing sequence that seems to be a temporal compression of the memory, if you like, for the previous experience that the animal has had. And there's a lot of evidence to suggest that this replay or reactivation plays an important role in memory consolidation. Okay, so what we want to know is whether activity during hippocampal shortwave ripples might actually extend beyond replay of direct experience to sort of facilitate the building of shortcuts in the cognitive map, of the building of inferred relationships between different memories. And so what we did was, again, we used a regression approach to try and identify neurons that preferentially represent the different cues in our task. And then what we asked was, how coactive are those neurons with one another during periods of rest and sleep? And so we identified periods where we see shortwave ripples in the hippocampus. And then we asked how coactive are our X, Y, and Z neurons with one another. And this is just an example triplet of neurons, but you can see that in late shortwave ripples, we see more coactivation of this particular triplet of neurons compared to early ripples. And that's not the case for this particular example of neurons that are representing set 2, the one that includes the neutral cue. As I'll show you, I'll quantify this across all our cells in a moment, but essentially during hippocampal shortwave ripples, there seems to be an increase in the simulation of the task model, if you like, the sort of structure of the task, and it seems that this seems to be prioritized for reward-related activity. So just to quantify this across all our triplets of neurons, here I'm plotting the difference between the coactivation of the X1, Y1, Z1 triplet during late versus early ripples. And you can see that for that particular or across all possible triplets in set 1, you see a significant increase in the representation of those triplets in shortwave ripples, and that's not the case for triplets in set 2. And you can compare that to a null distribution again, where we're going to shuffle the identity of the auditory cues. And again, this is significant across the population for the triplets in set 1, but not set 2. And then you might ask, well, okay, that's still sort of potentially just a representation of learned information. Is there also a change in the representation of X and Z without Y, which would be a stronger evidence for representation of an inferred relationship? And that is indeed what we found. So if you look at coactivations of X and Z without Y, for the set 1, the one that includes the reward, you see a significant increase in representation of that inferred relationship, but that's significantly different from cues in set 2, and also a sort of shuffled set of cues where you've got X2 with Z1, suggesting that this isn't just an increase in representation of reward, but it is in fact something linked to what's been learned on the task. So what this is showing is that there seems to be a representation of unobserved, and this is rewarding, relationships in the hippocampus during shockwave ripples in periods of rest and sleep. And we can think of this as potentially forming a shortcut for an inferred relationship. And just finally on this study, we also asked what's the temporal order of this firing of X and Z cells. And we found that there was a bias towards Z1 firing before X1 in these shockwave ripples. So the reward is represented first before the auditory cue. And that's consistent with quite a lot of the data from spatial navigation tasks, where you get reverse firing during replay in response or following receipt of reward. And so what this might be doing is potentially providing a mechanism for assigning credit to those auditory cues, which have never actually been experienced with reward directly. And so potentially the hippocampus might provide a sort of training signal to other areas like the neocortex and induce or facilitate the sort of synaptic plasticity in those other brain areas that to allow for representation of the inferred reward. Okay, but what was really challenging to look at in this particular study was the effect of these mechanisms that we're observing during shockwave ripples, these sort of replay-like mechanisms, on behavior. So we are looking, during shockwave ripples in our mice, but the behavior is a little bit noisy, and so trying to work out a relationship between what we observe in shockwave ripples and what we observe in behavior just wasn't possible. And so what we decided to do was to develop some studies in humans that would allow us to ask, does neural activity that happens during periods of rest actually influence subsequent behavior? And so I'm now going to turn to these two studies that look at try and address this question. And to do this, we used a method called or manipulation called targeted memory reactivation, or TMR. And this is a tool that can be used to try and boost memory reactivation and replay during periods of rest and sleep. So I'm first going to start by describing a study that tries to develop this tool, and then I'll show you how it has an effect on our ability to infer and build shortcuts in our cognitive map later on. So the idea here is really that we can try and use this as a manipulation to boost replay for certain memories, and use another set of memories that don't have this manipulation as a control, and then we can ask, for those where we've tried to boost replay, do we improve people's ability to infer? All right, so what is targeted memory reactivation? So this is a method that's used to try and facilitate the consolidation of cued memories by increasing the likelihood of replay of those memories during periods of rest and sleep, although I should say most of the literature is during periods of sleep. And there's a nice review article here if you're interested. So what happens is that during a particular experience, we pair that experience with a particular sound. So this is typically punctate cues that are used that are paired with particular spatial trajectories or particular stimuli presented to the rodent or human, typically. And then the idea is that you then replay those, I should use a different term, you then play those auditory cues back to the animal or the human during periods of rest and sleep. And then there is now evidence to show that this manipulation increases the likelihood of the cued memory being represented in the brain during rest and sleep. So effectively, you're increasing or facilitating replay of the cued memory in response to this manipulation. So this is, most of the literature has using this approach has been done during periods of sleep, particularly non-REM sleep, and most of it has also used punctate cues that have been paired with particular stimuli. So one of the really nice early studies demonstrating this was comparing the replay of left versus rightward trajectories in a rat during periods of non-REM sleep, and so they paired one of these two trajectories with a particular sound and then played the sound to the animals when they're in non-REM sleep, and found that the cued trajectory was preferentially replayed, or there was an increase in likelihood of that trajectory being replayed in non-REM sleep in those animals. But there's also a lot of human literature now supporting this idea. Okay, so TMR biases the content of replay towards cued content. And so now we're going to use this in the following way. So we decided to use our TMR in a slightly different way from what's been done previously. So we use contextual auditory cues. These are jingles, these sort of soundtracks that we're going to play to our subjects whilst they're learning particular associations. And we're going to also then apply that or play that soundtrack during a period of rest rather than in sleep. And I think the advantage of doing it this way is that it's very easy to use. So I guess we hope that potentially it might also be interesting to people working on translational approaches to TMR, where you might be able to boost, for example, rehabilitation in stroke, for example. In a way that's much more easy than trying to target particular phases of sleep at the end of, Okay, so the idea here is now that we have two memories, memory 1 and memory 2, and for one of these we're going to use targeted memory reactivation to try and boost replay, and then we're going to ask, does this improve inference? So are people more able to infer that Z, for example, was indirectly paired with X, compared to our control condition where perhaps people are still chaining together X to Y and Y to Z in the way that they learned it, rather than being able to infer that sort of shortcut. And so what we do is we have a learning session, and then we have a rest session, which is paired with TMR. So this cafe symbol is representing some kind of cafe soundtrack noises that we're using for our TMR protocol here. And we alternate periods of rest where people are awake but have their eyes closed, with periods where they're just doing a jigsaw to kind of keep them busy while we do the TMR. And then we have a testing phase at the end. Okay, so this is a study carried out by Annie Wilson, who's a very talented PhD student in my group, and Sumedha Nalaru, who was an RA and is about to start her DPhil here. And so we have two memories, as I said, memory 1 and memory 2, and I'll just walk you through what those memories actually consist of. So these diagrams might look slightly complicated, but essentially we get our subjects to associate each of these symbols with the one next door in a clockwise manner. And they do that for memory 1 and then memory 2. And each of these symbols is embedded within a background context. This is a room in a house. And each of these is unique, so they kind of move between the bedroom and, can't even see what that is, the kitchen maybe. So they navigate their way around the house. And we have a pink house and a green house. And each of these has a different soundtrack. So this one has sort of cafe background noises, and this one has jungle background noises. So while they're learning, they have those soundtracks playing. And so this is just an example trial from the learning periods that we use 3 alternative forced choice. So we show them a symbol in their room, and then they have to select from 3 options which symbol is in the next door, the neighboring room. And this allows them to navigate around the house and learn which symbols are paired together. On the bottom, you can just see their learning accuracy, so they get better over time. And then we fully counterbalance which map is then TMR'd, so which one we then effectively play the soundtrack for in the rest period. And you can see that for both the TMR and the non-TMR map, there is high learning accuracy, and there is no significant difference in learning accuracy between those 2 maps. Okay, so then what happens is we do the learning, we then have the rest session, we play whichever soundtrack is selected for the TMR, And then we put them through a whole bunch of different tests, and I'm not going to go through them all in detail, but essentially we just try and test subject accuracy for every possible relationship on those two maps. So we look at their ability to remember the relationship between scenes and cues, cues and cues. And then we also look at contextual relationships, like, did this cue belong to the pink or the green house? And so then we ask, is there a difference in the accuracy for the map that was TMR'd compared to the non-TMR map? And across all tests, this is across 40 subjects, you can see that there is a significant increase in people's accuracy for the TMR'd map compared to the non-TMR'd map. So the difference is shown by this grey distribution here. And then we ask, is this actually happening on all of our tests or not? And we split our tests into those that are looking at associative relationships, so for example, cue to cue or scene to cue, and those that are looking at contextual relationships, so distinguishing between the two maps. And for the associative relationships, we see the significant effect of TMR, but we don't see that for our contextual relationships, where they're distinguishing between the two maps. So this is showing that this TMR manipulation is improving the accuracy for the associative, but not the contextual memories. And then we can also ask, you might have spotted that we've got this inference test here. We can ask, are people actually better at inferring after they've, for the TMR map compared to the non-TMR map? So our inference test here now is we probe with a cue here, for example, and then we ask them to infer the cue that is 2 steps away on their map. And we find that there is indeed an improvement in accuracy for the map that is TMR'd compared to the non-TMR'd when they're performing this 2-step inference. So this suggests that TMR is improving behavioral accuracy for inferred relationships. And so this raises a couple of questions. First of which is, does TMR actually facilitate memory reactivation or replay? And secondly, does TMR during rest facilitate the formation of shortcuts in the cognitive map, in the underlying cognitive map? Okay, so to tackle this first one, like what's essentially going on in the brain during TMR, Annie performed this very heroic study on the 70 scanner where we did a very similar protocol to before, but now scanned our subjects while they're undergoing that rest period. And so for this study we used, it's very similar, but we just have 2 slightly simplified maps with just 7 symbols on each map, and we just changed the second map so that now it includes these faces rather than scenes. So we have 1 map with scenes and 1 with faces, and that's there to allow us to potentially look at differences in sort of bulk reactivation in response to the 2 maps across, for example, PPA and FFA. Okay, so we're now going to ask, can we see a bias in reactivation for the TMR'd map, and can we see evidence for sort of sequential reactivation of the learned information, so the associations between the cues during periods of rest? And so this is really building on an approach developed by Nico Shook and Yael Niv, sorry, there should be another C there, where they showed in the science paper that you can train a classifier to essentially distinguish between different representations in the brain, and then you can apply that classifier to periods of rest and try and establish whether or what's being reactivated in the sleeping or resting brain. So in our scan session, we've got our subjects lying in the scanner. They first perform this classifier training task, where we ask subjects to look at this scene, or they see a picture of the person, if it's the other map, and they have to vividly imagine the corresponding cue that goes there. And this is a task really designed to engage the hippocampus. And then on some probe trials, we test whether they got it right or not by showing a cue, and then they have to say, was it the right cue or not? So this gives us a sort of template representation for each of the 14 different nodes that we've got in our map, so 7 on one map and 7 on the other. And then we use our data from this task, our fMRI data, to train a classifier using leave one out cross-validation. And then we apply our classifier to the rest scan, where we have our soundtrack for the chosen TMR'd map playing, and then we take our scanned volume at each TR and we ask which of the 14 visual cues is most likely to be represented during that TR. So essentially, for each TR, we're saying, is it most likely to be Q1 to 14? And so the question is the TMR map preferentially reactivated during rest? And this is very preliminary evidence from a very talented MSc student who just finished her MSc in the lab. And she developed, Dennis developed a deep neural network approach to building the classifier, and compared that to using logistic regression and an SVM, and showed that for the data in the hippocampus, the deep neural network outperformed the other two methods, and same goes for data acquired in the visual cortex. So we then went with using this deep neural network to try and decode our 14 different neural representations, and then applied the classifier to our rest data. So what we found was, first we just looked at a kind of crude analysis. Are we more likely to decode the TMR'd map compared to the non-TMR'd map during our rest scan? This is a 20-minute rest scan, I should add. So what you can see here is the difference between our TMR and non-TMR map, and you can see that that is significantly different from 0, suggesting that the TMR is biasing the content of neural representations in the hippocampus during rest towards the map that is being cued. And then we might ask, okay, so are the learned associations, are they actually being reactivated in sequence? Is there sort of also evidence for replay, if you like? And so what we looked at here was the probability of observing one-length transitions. So these are the learned associations between our visual cues. And can we observe one-link transitions during rest above chance? So effectively, are we observing that we get representations of this cue followed by this cue? And are these kind of one-link transitions occurring more often than this cue to any other possible cue on the map? And what Denise found was that if you compare what's observed in the data to a null distribution where we're shuffling the classification across the 14 nodes, then you can see that there is a significant effect for this kind of sequentiality in the data. And also we tried this relative to a null distribution where we just shuffled within maps, so we kept those between map transitions, but shuffled within map which class was being, which node in the map was being classified. And again, in the data, we observed this significant effect where we're observing more one-link transitions in our rest data relative to the null distribution. So there seems to be some evidence for reactivation of sequential learned associations during periods of rest, and so we might tentatively think of this as a signal for replay. But of course, the temporal dynamics of fMRI data are very different from what we're observing with single unit spiking data in rodents. Okay, so now I'm going to go on and ask or does this TMR facilitate inference by building effectively a shortcut between X and Z in the underlying cognitive map? And again, we're just going to use behavior to try and adjust this. So we're going to use this TMR manipulation and again get our subjects to perform an inference task, and then we're going to try and use different behavioral readouts to assess whether we think there might be some evidence for a kind of shortcut being formed between X and Z in response to TMR. So this is a study by Shearer, who's a very talented DPhil student in my group. And the structure of the task is the same as the one I showed you at the start in the first study. So we've got auditory cues X paired with visual cues Y, and visual cues Y paired with monetary outcomes Z, and then we're playing the auditory cues and assessing evidence for inference. And just to confuse you, I'm going to change all the schematics, so the visual cues look like this, and the auditory cues look like this. And now we've got two contexts. So We've got one which is going to be with the cafe music, the cafe kind of soundtrack, and another one with the jungle, and this is to allow us to then use that TMR manipulation. All right, so the question is, does TMR facilitate inference by building a shortcut between X and Z? Yeah, okay. So to try and address this, we kind of try to tackle three questions. So first is the kind of replication of the previous data I showed you. Does TMR facilitate inference? Second is, does TMR impair flexible value updating? And I'm going to explain what I mean by that in just a moment. And the third is, does TMR build a direct association between X and Z? So we have a learning phase where they learn to associate auditory cues with visual cues, and then they do the conditioning, visual cue to outcome. And then we have a rest session where we're going to play the soundtrack for the TMR condition, and they're going to alternate between awake, rest with eyes closed, and doing a jigsaw puzzle. And then we're going to do a whole series of tests that allow us to try and tackle these three questions. So I'll go through these as I show you each bit of data, but effectively they're doing an inference test, and we're then going to do this, this sort of test of flexible value updating by changing some of the values of the visual cues, and then we're going to test inference again, and then a final test of inference right at the end, which I'll explain in just a moment. Okay, so the first question is, can we replicate our previous result and show that TMR facilitates inference? And indeed we can. So here we're playing the auditory cue, and then subjects are picking which of the outcomes they think is paired with that auditory cue. And in the TMR condition, people are more accurate compared to the non-TMR condition. Okay, so this is across 25 participants. So this is a replication showing that TMR facilitates inference. And then second question is, does TMR impair flexible value updating? So the idea here is that if we change the value that's assigned to the intermediary visual cue, does that affect people's ability to infer what value is, can be inferred from the auditory cue? And the idea is that if people have formed a shortcut between the auditory cue and the outcome, they may be actually impaired at updating that value when the visual cue has now got a different value assigned to it. Whereas if they are not, if they haven't got a shortcut between the auditory cue and the outcome, they may instead think of the auditory cue followed by the visual cue followed by the outcome. They may not be impaired at this change in value. So what we did was we got subjects, I'll just go back one slide, so we got subjects to learn that half of the visual cues had a different, sort of changed their values from, so if they were previously rewarded, they were now neutral, and vice versa. And then we got subjects to perform the inference test once more, and then we assessed whether they, how well they did in both the TMR and the non-TMR condition. And what we found was that in the TMR condition, there is a relative impairment on their ability to infer the appropriate outcome when the outcome paired with the visual cue has been flipped. And so, and you can see the difference here for the TMR versus the non-TMR condition is significant. So this suggests that value updating is relatively impaired for the TMR map, and so this suggests that perhaps people have formed a shortcut between X and Z, such that when the value of Y changes, the value of X, or the inferred value of X, is not able to be updated appropriately. And then the final test was, can we see any evidence for a direct association between X and Z in our subjects? And so what we're really comparing here is what I'm calling a shortcut X and Z versus a chaining of X to Y to Z. And to try and test this behaviorally, what we did was we played our sound now with the outcomes on the screen, but also with the different visual cues on the screen. And this happened right at the end of all the testing. And then what we did was we used eye tracking data to look at how much time people spent looking at the visual cues while they had to make a decision between which outcome was paired with that auditory cue that had been probed. And what we found was that in the non-TMR group, people spent more time looking at those visual cues than in the TMR condition, suggesting that perhaps people have formed a shortcut between X and Z when they've received TMR compared to when they haven't. And so when they haven't received TMR, they still rely on that intermediary visual cue in order to make the inference. Okay, so just to conclude, so I've shown you 3 studies, and I've just put the publication references here. The first, I showed you that in the hippocampus, during periods of rest and sleep, we see evidence for the representation of inferred relationships. But during inferential choice, it's just the learned associations, the directly learned associations that seem to be represented. And then I showed that we can use targeted memory reactivation to boost memory reactivation and replay, and we can use that manipulation to also facilitate inference. And I've shown you some behavioral evidence to suggest that perhaps this TMR manipulation might also facilitate the building of a cognitive shortcut. And with that, I'd like to thank everybody who did the work. So Annie and Sumedha carried out the second study with the behavioral study, and Annie also ran the fMRI study with some work by Dennis for the analysis of applying the deep neural network for the classification. And Cal ran the final study that I showed you, the behavioral study. I'd also like to thank David Dupre, who supervised the first project I showed you, and thank Jill, who co-supervised the final 2 studies. And thank you all very much for listening.
Testing. There we go. All right. We'll take a few questions. We'll do it the same way from the stage and then on the slides. So I'll start here, and then we'll go there. Go ahead.
Thank you for the talk. So I have a kind of a general question, which might be related to more than your talk and might be annoying a bit. And the question is the ratio between the huge variability between subjects, the differences that you show between the means. So you draw conclusions from the means. You say TMR improves representation or something like that. But the variability between subjects is huge. I mean, many subjects show decrease, many subjects show increase. The variance is some 10 times more than the difference between the means. So I'm interested to know what you make out of it when you suggest, you say, yes, it can improve, but you know that at least half or close to half of the subjects, it does not improve the representation. So what would you make out of this?
Yeah, I mean, I think certainly if we want to think of it as like a translational tool, then we need to take into account the variance and take it seriously. Yeah, I mean, we don't know, right, what causes the variance, whether it's the time of day. We try to test people at the same time every day, but not always. Whether it's, in the first study, we've done some careful regression analyses where we've controlled for sex and age and, which map was TMR'd, for example. And none of, if you take into account all of those factors, you still get the same results out. So we don't think that any of those are explaining the variance. So I don't have a good answer for you as to, what's causing it. But certainly, if we want to use it as a method translationally, and which people are trying to do or trying to start doing, then we need to try and understand what it is that...
So you treat the variance as a noise that you cannot explain, but not as a real thing, if I get it right.
It may well be, something that we just haven't understood yet. So some factor that we haven't measured or, yeah.
Thank you. All right, over there. Oh, sorry, we'll do it then.
Thank you for the talk. Can you hear me okay?
Yeah.
Yeah, so as I was listening to your talk, as I was building memories of what you're talking about, I was always wondering how was my model of the brain being updated in the neocortex, right? So my question is, how does this information, these memories, this perspective coding you're building in the hippocampus, then becomes consolidated in the neocortex? In particular, given that the experiences are no longer there, right? So how does the brain know which brain areas to change? Yeah.
Very good question. I wish I had the answer for that. Yeah, so I didn't show you today some data where we're trying to kind of tackle that, where we're recording simultaneously in hippocampus and neocortical regions. And yeah, I think it's a kind of big unsolved question at the moment, how a particular replay trajectory, whether that's through space or abstract cognitive map in the hippocampus, might translate into strengthening of synaptic connections in the specific neurons that are relevant for the memory that are distributed across the whole of neocortex potentially. Yeah, so I think we can think of, in a way, the hippocampus as representing like a pointer or a kind of index for those neurons that are represented in neocortex, but how it does that we don't know, whether that's just through anatomical connections or whether it's something more complicated. But certainly we seem to see, so I didn't show it today, but we seem to see coordinated reactivations in like visual cortical areas, for example, in response to the hippocampal activity that we observe on our inference tasks. So there's certainly coordination going on. How that coordination is made specific, I don't know.
Go ahead.
Hi. Great talk. I was wondering if you've looked into whether there's any interference between the memories for the TMR context and the non-TMR context. So specifically, you see that there is this difference whenever you have the TMR, but is that how much of that is driven by an increase in memory for the context that you have TMR versus maybe a decrease in memory and consolidation for the context that is not?
Yeah, that's an important question because I think, so the sort of overall consensus in the TMR literature is that the non-TMR condition actually goes below what you would expect without TMR. So we haven't controlled for that. So we see a difference between the two, but we can't say explicitly whether we've got an increase for the TMR and versus a decrease for the non-TMR.
All right, I think we'll stop there and let you guys head to lunch. Please join me in thanking Helen one more time.